So now that we've seen Mendel's experiments, we're going to talk about what's going on behind the scenes, controlling the genotype that's in there. And we also need to look at what happens when we have multiple traits that are in there. So we'll talk about Mendel's law of segregation and independent assortment in terms of genetics and what's going on in meiosis. We will examine this thing called the fork line method that can be used to calculate genotype and phenotype probabilities. This is based on our probability laws that we've talked about. We need to talk about this thing called linkage and recombination on gametes that are made in the genotypes that are in there. And then we'll talk about this process called epistasis, which can affect a few different things. So Mendel did all this work, and he did not know what the genetic material was. He didn't know about DNA, didn't know about chromosomes. This is all pre those discoveries. So it's really amazing that he was able to put everything down into a couple of rules, and he created what we call 4 postulates here. They're basically rules of genetics, and the rules summarize most of his what we call classical genetics. They don't handle every situation because he didn't know how this was actually occurring. He couldn't predict everything. So there are some caveats here. That's because Mendel didn't know about DNA or chromosomes. And it's actually interesting. Mendel's work was published, but not widely distributed. So much later, other people kind of rediscovered his findings and rediscovered his work. So he wasn't really recognized for his discoveries in his lifetime. So Mendel's laws of inheritance. Well, first off, his first postulate here. There are pairs of unit factors in there. We call these genes. Each organism has a pair of hereditary factors, and each parent passes on one to the offspring. These are discrete units. So we saw that in the genotype, right? There was one allele from one parent, one allele from the other. In the organism, you're diploid, so you have 2 of each of these things. And there are pairs of these alleles or genes in there. Alleles can be dominant or recessive. This is the law of dominance. In a heterozygote, one form will conceal the other, so the dominant will mask the recessive. So only one allele contributes to the phenotype. And in order to show the recessive phenotype, you must be homozygous recessive. It's not always this simple. This is one of the places where Mendel didn't have all the answers. Number 3, equal segregation of alleles. The law of segregations. Genes must segregate equally into gametes. Offspring need to have an equal chance of inheriting each different allele. So our Punnett square shows us equal probability, and we get that 1 to 2 to 1 genotype ratio in the F2 because there's an equal chance of getting either of the alleles that come from a parent. So there's no bias in what gets passed on. Our final one, which we need to examine more closely here in a second, is the law of independent assortment. Independent assortment, genes do not influence the inheritance of one another. All gametes with combinations of alleles are equally possible. So to understand this, we need to look at a dihybrid cross where we have 2 traits. This is essentially saying my height does not affect the transmission of my eye color or something like that. And for Mendel's pea plants, for the traits he was looking at, this is true. But there are some big exceptions to this one that Mendel could not predict. So let's take a look at what's going on here. So here's the law of equal segregation and its basis in meiosis. So in meiosis, right, we have the chromosomes pairing up. Here we have the sister chromatids, the homologous chromosomes paired up, and the homologous chromosomes get pulled apart. What equal segregation is saying is that getting a big R is equally likely as getting a little r for this gene, right? It's not like, 3 of the gametes get big R's and only 1 gets little r. No, there's an equal chance of big R being there. But you'll see that there are more than one gene being looked at in here. So we need to take this a little bit further and look at what's going on here. So let's talk about a dihybrid cross. We're not going to go too deep on this. You'll do that in 300 level genetics, but a dihybrid cross looks at two traits. In this case, we are looking at seed color and seed shape here. So let's look at the two versions of each of these. We have yellow seed color and green seed color, and we have round seed shape and wrinkled seed shape. There's a couple of important things I'll show you in a moment, but let's look at what happens when we do this dihybrid cross. So we take a yellow round parent and cross it with a green wrinkled parent. Okay, here's an important thing. We're using Y for yellow and little y for green. Don't put a G in there, right? That'll confuse yourself. Same thing with round is big R, wrinkled is little r, okay? So don't get confused there. Okay, so we cross these two and we look at the F1. So what does the F1 show us? It shows us all the same phenotype. And what did our monohybrid cross tell us about that phenotype? Well, just like the monohybrid cross, the phenotype shown in the F1 is the dominant phenotype. So the organism is heterozygous for both of these genes, and it shows the dominant phenotype. In this case, yellow and round. So our phenotype that is dominant is the yellow seed and the round seed shape. Okay, that means wrinkled and green are recessive. You could already kind of see that because I've spoiled it with these things. But let's look at what happens when we do the F2 though. We self the F1s, and now it gets a little bit more messy because we have more squares in our Punnett square, because instead of just one gene, we have two genes, and we need to work out the possible combinations here. Okay, you could add all these up, but what you will find is that there are 9 yellow round seeds for every 3 green round seeds, for every 3 yellow wrinkled seeds, to every 1 wrinkled and green seed. So there is 16 squares here. There's a 9 in 16 chance of being this. There's a 3 in 16 chance of being that, 3 in 16 chance of being that, and a 1 in 16 chance of being that. What I want you to take from this is the F2 dihybrid phenotype ratio, which is 9 dominant to 3 dominant recessive to 3 recessive dominant, that's reciprocal, to 1 recessive. Regardless of what the trait is, it always shows up this way. So you can see that pattern here. Now, drawing this all out and writing this all out, all these combinations, is kind of a pain. So you could use the product rule that we saw in the last section and do the two kind of monohybrid crosses on their own. So what's the probability of getting a round seed and yellow seed color? Well, for round, we know it's dominant, so there's a 3/4 probability of that. And using the product rule, we multiply that by a 3/4 chance of getting a yellow seed color, which is also dominant. So we end up with 9/16. I think you will find that that is what we observed counting out the Punnett square. So essentially, it's 2 3-to-1 ratios intermixed in there. Okay, so why does this happen? Why is it evenly distributed? Why isn't it that green and wrinkled are somehow interacting with one another? Well, this comes down to the chromosomal basis for independent assortment. In this case, we have our gene for seed shape, R, round and wrinkled, and our gene for seed color, yellow or green, yellow, green. So during meiosis, these two genes assort independently because Mendel was really, really lucky. Here you can see the R's segregate here and the Y's segregate here. So seed shape and seed color are assorting independently, and you have an equal probability of getting a little r and a little y, as a big R and a big Y, as a little r, big Y, or a big R, little y. Equal chance here because of how metaphase is working in here. This is independent assortment, but there's a big caveat here. Can you think up of an exception to this rule for independent assortment? What would cause this not to be the case? Go and pause the video, think about that. All right, well, Mendel got lucky, like I said, because his two traits were genes on separate chromosomes. What would happen if these genes were on the same chromosome? Things might get a little bit different there. And that's where we get the concept of linkage. So linked genes can violate this law of independent assortment. So if genes for traits are located on the same chromosome, they won't necessarily undergo independent assortment. They're coming together because they're on the same thing. So we instead can consider the possibility of recombination, though, to create new combos. But recombination does not happen every single time. The closer two genes are, the less likely that recombination is to occur. So linkage is segregation influenced by genes located physically close to one another on the same chromosome. The closer they are, the more likely they will not recombine and will instead be inherited together. And there's complicated calculations you can do to actually calculate how close they are based on crosses. That's all 300 level genetic stuff. So look forward to that. But let's look at what we're talking about here. We have big A, big B, and big C alleles all on this chromosome, and little a, little b, little c combinations on that chromosome. If recombination occurred, we could get a big A, big B, little c, and a, little b, big C. Okay, but there are non-recombinants on the outside. So if recombination occurred between B and C, we could see it at most 50% of the time. So this comes to the idea of parental types versus recombinant types, and we need to clarify that. Let's go back to our previous cross here. So parental versus recombinant types. Let's assume these genes were linked. Okay, I know I just showed you they weren't, but let's assume they were. What is the parental combination of traits in here? Well, that's the combination of traits found in the parents of a dihybrid cross. Makes sense, right? So in this case, we would have yellow and round seed, color and shape. That's one of the parental combinations. Yellow seed color, round seed shape. The other parental type is green seed color with wrinkled seed shape. Okay, I think that's simple enough. That's what the parents looked like. All right, now here's the question. What would the recombinant types be? Go and pause the video. Think about that. All right, well, they have to be different from the parental types, right? And there's only two possibilities here. So recombinant types are new combinations of traits not observed in the parental generation. So in our case, we would have green seed color with round seed shape. That doesn't look like either of our parents. And yellow seed color and wrinkled seed shape. Now, if we were independently assorting, we said there was an equal probability of getting this, or this type in there. We should see that, 50% are recombinant, 50% are parental type. All right, so let's look at an example here. I have some flies, and I need you to figure out what the parental types and the recombinant types are. So here are the two parents, and here are the offspring. My traits are body color and wing size. Go ahead and pause the video, look at them, and tell me what the parental combinations and what the recombinant combinations are. All right, so for this example, our parental types would be gray body with long wings or black body with short wings. Those are our parental types. That means our recombinant types would be gray body with short wings or black body with long wings. And in this case, they are independently assorting because the genes are on different chromosomes. So there's no linkage, they're unlinked. We get an equal probability of all of the different gamete types. Here, we could get big G, or little g, little l. We could get big G, little l, or little g, big L, all in equal combinations here. But let's look at what happens with linkage. So here's our unlinked example when they're on separate chromosomes. Well, there's a couple of different possibilities that could happen here, depending on where the genes are. If genes are really, really close together, like super close, the probability of crossing over is very unlikely. And let's say, just for hypothetical reasons, that no recombination ever occurred. In this case, we could only ever get the parental combinations of alleles, right? Big G, big L, and little g, little l. So in this case, we would only ever get offspring that look like the parents. Gray body, long wings, black body, short wings, right? None of the recombinant types could occur because no recombination has occurred. What if crossing over happened 100% of the time? This might seem like a hypothetical, but actually when genes are on opposite sides of the chromosome, opposite sides of the centromere bit of it, they do effectively recombine 100% of the time. Well, then they would look like they were independently assorting, and we would never know. So we would get 2 parental types and 2 recombinant types in equal proportions here. But what if they're kind of close together? And this is what Thomas Hunt Morgan, who did a lot of genetic work on flies, actually observed. Well, he observed that in this case, there were a lot more parental types than recombinant types. In fact, it was about a 1:1 to 0.2 to 0.2 ratio. So he had about 1,000 of the parental types and only about 200 of the recombinant types. Well, what does that indicate to us? That indicates that the genes are close together. They are linked together. So what I want you to take from this is that gene linkage will show deviations in the ratio of expected recombinants to parental type. We will see way more parental type when genes are linked than we see recombinant types. Okay, that's what we're taking from this. Don't worry about the numbers of the calculations or things like that. What if you have more than 2 traits? Well, the Punnett square would get really ugly, right? Because then that's a lot of square. I think it's like 64 squares at that point. What you can do, though, is use this fork-lined method where you use probabilities, right? So for a trihybrid cross, you could look at each as an independent probability, 3/4 for dominant, 1/4 recessive, and you could work through. In this case, we're looking at seed color, seed shape, and plant height. So if I wanted to say, what's the probability of getting a green seeded, round seed with a dwarf plant, right? I would go, well, green is recessive, so there's a 1/4 chance there. Round is dominant, 3/4 chance there, 1/4 times 3/4, and then dwarf is 1/4, so I would multiply that by 1/4, and that would be whatever that value comes out to be. You can figure out, here's the F2 ratio, but you can work this out through the probabilities there. We're not going to do that. That's a 300 level genetics thing, but just showing you that these are independent events. We can use probabilities on them. All right. Another problem here is that how genes actually function to make the phenotype happen is more complicated than Mendel could understand. So there is a concept called epistasis. This is where the expression of one gene can mask the expression of another gene. Epistasis literally means standing upon, so that's kind of what it's getting at, right? So not all traits are controlled by a single gene. For like human eye color, there's at least 8 genes with multiple alleles in there that control it. So that's why we don't get like white or black eyes or something like that, right? Like there's a lot of different variation in there. In mice, this agouti gene is dominant to solid fur color. This agouti coloration is dominant to solid fur color. Okay, but there's a second gene needed for pigment to even be produced. So the second gene can be what we call epistatic to this. And we see that even though if you have 2 dominant agouti genes or even 1 dominant agouti allele, it can be masked by the recessive pigmentation production gene. These recessives become albino. So it screws up this patterning, really. We should see, in this case, for the agouti and the solid fur color, we should see 3/4 this and 1/4 that, but we don't quite see that ratio here. The albinism becomes the kind of confounding factor because it can mask the expression of this. Again, I'm not worried about the phenotype ratio or anything like that that's occurring here. What I am worried about is that you understand that one gene can affect the expression of another, and that you understand that's called epistasis, that it will be explored more in 300 level genetics as well. Also, in my micro 250 class, we go into this a bit deeper, somewhere in between this class and 300 level genetics. So if you find this stuff really, really cool, my micro 250 class is the place for you. Okay, so let's do a review question before we finish up here. You've performed a dihybrid cross on a new species of flowering plant you discovered. Okay, your dream, right? You examine the segregation of flower color and plant height. You have two flower colors, yellow and white, and two plant heights, tall or short. So you take a tall white parent plant and cross it with a short yellow parent plant, okay? So you would expect a 9:3 to 3:1 phenotype ratio in the F2, right? But your crosses get results that deviate significantly from this. You mainly see tall plants with white flowers and short plants with yellow flowers. What do you hypothesize has occurred here? Go and pause the video and think about that. All right, well, hopefully when you saw that the ratio deviated from the expected results, you thought gene linkage, right? In this case, it's most likely that the gene for flower color is linked to the gene for plant height, and that's why it's deviating from that ratio. All right, so let's wrap up here. Mendel used his data from all of his experiments that he did and formed several rules or laws of inheritance. First off, there were pairs of unit factors. Genes are discrete units. Each parent has two alleles and gives one allele to the offspring. We have the law of dominance. Some alleles are dominant, and they will mask the recessive alleles. For recessive, you need two copies to show that recessive phenotype. The law of segregation. Genes segregate equally into the gametes. You should get in a monohybrid cross a 1:2:1 genotype ratio. There's no favoring certain alleles over one another. Law of independent assortment. Different genes do not influence the inheritance of one another. That was Mendel got lucky because all 7 of his traits happened to be on the 7 different pea plant chromosomes that he had. So he did not run into the exception, which is if genes are linked on the same chromosome, which then will kind of destroy independent assortment. But we know why, right? It's not magic. We understand why that occurs. So we looked at a dihybrid cross for 2 unlinked traits. In the F2, we should see a 9:3 to 3:1 phenotype ratio. Remember, the F1 was the same, shows the dominant phenotype, all heterozygotes. That F2 phenotype ratio, 9 dominant to 3 dominant recessive to 3 recessive dominant to 1 recessive recessive. That's our phenotype ratio. Deviation from this could likely indicate linkage, possibly epistasis, but in most cases it is indeed linkage that is the cause of this. Linkage is when 2 genes are located close together on the same chromosome. Only recombination can form new combinations of alleles in this case. Epistasis, as we talked about, is where the expression of one gene impacts the expression of another. So if you don't have the gene that allows any color to be made, doesn't matter what your genotype is for the other color determiner, you have no color at all, right? So that is it for chapter 12.3. We're going to continue on, and now we're going to start to incorporate our molecular understanding of genetics into Mendel's understanding, what we call classical genetics.